Hello, my dear students. Welcome back to another learning lessons in my Physio-Pulse channels. Today we will discuss another metabolic pathway, that is urea cycle, very important for your examination purposes. And we will discuss here very simple way, easy to understand, and you will find the clear explanations, diagram, important concepts here. Before we begin, if you find this video helpful, please like, share, and support the channel. And now let us understand about the urea cycle. In 1932, the Urea cycle is the first metabolic pathway which was elucidated by Sir Hans Krebs and also Henseleit. So this cycle is called the Krebs-Henseleit cycle. As ornithine is the first product of this cycle, so it is called the sometimes ornithine cycle. Actually, ammonia is high toxic to the central nervous systems and it is converted to non-toxic urea in the liver only. Urea is water soluble, easily excreted by the kidney in the urine, and urea is the main end product of the protein, like the amino acids metabolism. Plasma urea is 15 to 45 milligram per deciliter, and it is formed in the liver and transported in the blood to the kidney to be excreted in the urine. So, urinary urea is, that is 25 to 30 gram per day. Urea cycle occurs in 5 stages, or 5 steps. That is, the first steps involved in mitochondria, and the last 3 steps that will actually metabolize in the cytoplasm. So it utilizes 3 ATPs and 4 high-energy phosphate bonds. The 5 steps catalyzed by 5 enzymes, and any defect of these enzymes leads to ammonia intoxications. So let us understand how this cycle or how the steps occur in the urea cytoplasm as well as the mitochondria. In this urea cycle, there are 5 steps already. We told that was the 2 steps, that is the number 1 and number 2 steps that occurs in the mitochondrial. And in these 2 steps, there are the 2 enzymes important. One is the CPS-I, carbamoyl phosphate synthetase 1, and ornithine transcarbamylase enzyme, which occurs in the mitochondria. And another rest 3 steps that was specifically argininosuccinate synthetase and argininosuccinate lyase enzyme and arginase, in these 3 enzymes which are involved in the cytoplasm. So if you see the whole reactions, whole cycle, there will see these are the 5 enzymes. So already we told these are the 5 enzymes, and first 2 enzymes, that is in number 1, when ammonia combined with the carbon dioxide to form the carbamoyl phosphates with the help of carbamoyl phosphate synthetase enzyme, that is CPS-I. And here the 2 ATP which is converted to form the 2 ADP plus PI. Now in the second step, what happened? Carbamoyl phosphate combined with the ornithine and to form the citrulline. So in these 2 steps occurs in the mitochondria. And here the enzyme is ornithine transcarbamylase. Now this citrulline combined with the aspartic acids and to form the argininosuccinate with the help of argininosuccinate synthetase enzyme. Earlier we have discussed that synthetase means where the ATP was used, that is ATP is converted to form the AMP plus PPI, and actually 1 molecule of water is removed in these reactions. And now argininosuccinate, which is converted to arginine, and succinic acids, which is converted to fumaric acids. So arginine, with this help of the enzyme, that is called the argininosuccinate lyase, because it can break down, so it is lyase enzyme. And this fumaric acid goes to the TCA cycle, and ultimately it forms the urea cycle, and aspartic acid is again formed, and in this way this cycle continues. Now arginine, what happened? It will combine with the arginase enzyme and to form the urea and 1 molecule of ornithine. So in this way this cycle is continuing. So urea cycle steps, if you see, mitochondrial reaction, carbamoyl phosphate synthetase, the key enzyme of this urea cycle, and this synthesis of carbamoyl phosphate from the ammonia and carbon dioxide ATP already discussed. And second ATP molecule is required to supply the energy of this reaction, and N-acetyl glutamate is an activator of this enzyme. In the mitochondrial second step, that is the ornithine transcarbamylase. This transcarbamylase phosphate groups with ornithine to form the citrulline, which is transferred to the cytosol. Now it goes to the cytosol. Now in cytoplasm, first citrulline unites with the aspartic acids in presence of the argininosuccinate synthase, and to form the argininosuccinate acids. We have the two high energy phosphate bonds are hydrolyzed to provide the energy for the reactions. And this argininosuccinate is converted to arginine and fumaric acids with the help of argininosuccinase or lyase enzyme. Then again this arginine by the arginase enzyme, it is hydrolyzed to form the urea and one molecule of ornithine, and that repeats the cycle. So already two molecules of ammonia present in the urea, and one is derived from the carbamoyl phosphates, another is from the aspartic acids. So in this way urea cycle is happening. And now if you see, the urea cycle is called the bicycle because of that urea cycle in the first, if you see urea cycle when it is running, at the same time there the TCA cycle also running, because argininosuccinate it will lyase to form the arginine and 5 fumaric acids, and this fumaric acid converted to form the malic acids, then oxaloacetic acids. Already we discussed in the TCA cycle, and this oxaloacetic acids it will convert to form the aspartic acids. Again it comes to the argininosuccinate. So in this way this bicycle is happening. And now what are the important actually diagnostic importance of the urea concentration in the blood? Actually, if you see, plasma urea is one of the key important functions, kidney functions test. And plasma urea, if it is increased in the kidney, the disease that like the renal failure or uremia is happen. In liver failure, liver cells cannot convert ammonia into the urea, so there will be hyperammonemia. That means ammonia intoxication is started and urea is decreased. So relationship urea cycle and TCA cycle, fumaric acid produced urea cycle by oxidized to Krebs cycle, oxaloacetate, that we already discussed in the bicycle form. And this is the cycle of urea cycle where carbon dioxide or H2O, NH3 and ATP combined CPS-I to form the carbamoyl phosphates, then ornithine transcarbamylase combined with the ornithine from the citrulline, and this has happened in the mitochondria. But in the last subsequent steps, it will form in the cytoplasm, that we will discuss here. Now, one of the important diagnostic or disease is that hyperammonemia. When the normal level of ammonia in the blood, which is 0.5 to 1 mg per deciliter, if it is increased, then hyperammonemia occurs. And these are the symptoms of the hyperammonemia: lethargy, coma, even also growth retardation and hypothermia, even poor coordinations, like this ataxia and seizures, and these are happening into the central nervous system, general, muscular, even eyes, pulmonary, liver, everywhere it is seen into these symptoms, hyperammonemia. And there are the various types of hyperammonemia and causes is there. Acquired hyperammonemia, which is the liver failure due to the impaired urea synthesis, renal failure due to the impaired excretion of the urea and ammonia, and shunt operations between the portal and systemic circulations, and collaterals between the portal and systemic circulation due to the liver cirrhosis by the this is hepatitis. And now another type is genetic defect in urea cycle enzyme. If it is happened, then hyperammonemia happened, and this is happened type 1, type 2. These are the hyperammonemia, or even also citrullemia, which leads to citrullemia. That means the citrulline level increases into the blood, and even also these are the happening into the during the genetic defects in urea cycles. And type 1, type 2 hyperammonemia are more severe than the other types. And the high serum ammonia produces the brain damage. What happened? Actually, ammonia when it increases, it leads to the it leads to the damage of the brain, as ammonia will combine with the alpha ketoglutarate acids and forming glutamate or glutamine, and this will decrease in the energy productions of the Krebs cycle in the brain, so leading to the brain damage. So it is manifested by the irritability, tremors, vomiting, and blurring of the visions, and inverted sleep rhythm, coma, etc., and it will lead to the mental retardations. Also, ammonia causes the alkalosis. So the treatment is very important. The treatment is by a diet which is containing the low protein diet. So this is all about the urea cycle, and I think we have covered the steps of urea cycle as well as the diagnosis or symptoms of the urea cycle disease, hyperammonemia. And one important thing we also discuss here: when the high serum level produces in the brain, then brain will lead to the damage. So in this way, a urea cycle is very important for practical cases. and hyperammonemia, which leads to the which leads to the brain death. And already we discussed here. And one important thing we also highlighted here, that is particularly the energetics of urea cycle. Actually, if you see the overall cycle, NH3 plus CO2 plus aspartic acids, which will form the urea as well as the fumaric acids. So in urea cycle, 2 ATP are used in the first reaction. Already we discussed 2 ATP are used in the first reactions, 2 ATP used in the first reactions, and this another ATP is converted to AMP and PI, and which is equivalent to 2 ATP. This is AMP plus PI, and total 4 molecules of high energy phosphate bonds are also found here. But one important thing, fumaric acid is formed in the fourth step. Fumaric acid is formed and may be converted to the malate, and malate oxidized to the produced to the oxalate states. So in here that the 1 NADPH is required. We have already learned about in this pathway in the TCA cycle, and where 2 molecule of ATP, 2.5 molecule of ATP will be produced. So net high energy expenditure is 4 minus 2.5, that is 1.5 high energy phosphate bonds. So urea cycle and TCA cycle, they are both interlinked, so it is called the urea Y cycle. So students, we already covered everything in your exam purposes of urea cycle. So if you found this lectures helpful, do not forget to like, share and subscribe to the channel. Press the notification bell so you never miss of our upcoming biology, physiology or biochemistry lectures. And thank you for watching, and we'll see you again in our next